In terms of actual new stuff to put in your lab book, right now, today will be the very last new stuff that you put in there. In terms of the actual lab itself, I've been checking so at this point, it looks like most people really do have a pretty good sense of what we're doing here or what is going on. In all cases, we are looking at how the relationship of like the enzyme and typically pH, if we're looking at part 1 or part 2, or else the enzyme and the presence or absence of bile in part 3, affects the breakdown of either carbohydrates, proteins, or fat. So again, for part 1, which tube do you think is going to have the best breakdown of starch? I don't hear anybody.
Tube 2.
Tube 2. And why is that?
Because of the saliva enzyme.
Right, so we have the salivary amylase. And then what is the other thing what other condition is there that is going to make it have the best breakdown compared to the other tubes?
pH closer to 7.
Yeah, it's in the right pH zone, right? Salivary amylase is going to be optimized to work near neutral because it just is in your mouth where it's not too basic or too acidic. So tube 3 also has saliva and starch, but it's really low pH. And what happens if you take a protein and put it in a pH that's not what it likes to be in?
It'll denature.
It denatures. It changes its shape. And when it changes its shape, the active site will no longer bind the substrate, and it's no longer going to do its job. Right? That's going to be the same kind of thinking that when we get down to the protein one. The protein one, we have egg white and we have pepsin. Pepsin is this enzyme that breaks down proteins. What pH does pepsin like to be in?
A pH of 2.
Yeah, exactly. It wants to be in a really acidic environment. That's what it's optimized for. So in this case, we're going to have more neutral in tube 1. We'll have it in a more acidic environment in tube 3 and a basic alkaline environment in tube 4. So when you're like thinking about which of those tubes is going to have the best protein digestion, theoretically?
Tube 3.
Yeah, tube 3. Because that's the one where we have the pH closest to the pH that the enzyme is optimized to work in. So, when you're, takeaways from this, some people said the right thing in the prediction, but they didn't kind of take it to that level of being very clear, like, tube 4, it's alkaline, and that is really not what pepsin likes. It's going to denature it because pepsin is actually optimized to have the right shape when it's in an acidic environment. Tube 1, it's probably not going to be in its optimal conformation either because it's not as bad as being an alkaline, but it's not going to be what it really likes. What about the egg white and just in acid? So this is really weak acid. This is like diluted vinegar, basically. This is not like, obviously, if you take some concentrated acid and put something in there for 6 hours, it's going to start hydrolyzing the bonds and stuff. But if you have pickles, cucumbers in vinegar and put them in the refrigerator, or pickled eggs for that matter, they can be in the refrigerator for months and it doesn't turn the stuff into, doesn't dissolve it, right? Like a dilute acid sitting for a little while is really, it's going to denature things. It's going to mess with the folding of the egg white albumin, but it's not going to break it apart. What do we need to actually break the peptide bonds that hold the amino acids together?
Enzymes.
Say it again.
Enzymes.
Exactly, the enzymes. So tube 2, it's got the right pH that we're looking for, but having it acidic is not going to really break anything down. We need it to be acidic, and then having the enzyme that also is optimized to work in that environment to really have some breakdown going on theoretically. All right. Are there any questions about any of the hypotheses? Most people seem like they had a pretty good sense of what's going on. All right, so what we're going to do now is, again, it's not as much fun for you. It was probably more fun for us to film these things, but I do think it is important to kind of have a more physical sense of what these experiments look like, just so you see what the data looks like, and also so you can get a sense of the things that can go wrong. And I might speed up through some of the stuff to make sure we don't run out of time, but this should just work out perfect. So here we go. Wait, what's going on?
Okay, we're going to be going over part 1, salivary digestion of carbohydrates. And the enzyme we're using for this part of the digestive lab is salivary amylase. And the source of that is our saliva. So it says to collect 7 mils of your own saliva in a graduated cylinder. So I have close to 8.5, almost 9 mils. And so I've been elevating, collecting it in my little spittoon graduated cylinder.
Right, so partly just to show you, it really is like in the data that you're going to see, it's Becky's spit. This isn't like just made up, like, oh, theoretically. When you see the results that are happening, it's from some factor that actually is in Becky's saliva. So that's why we're kind of making sure you see this, since obviously you're not actually spitting and watching your own spit do the magic.
We're going to be using this for not only the spot plate experiment, but also for the 5 tubes of the carbohydrate digestion.
So here again is my lab partner saliva that we were using as our source of the amylase enzyme for the carbohydrate part of today's lab. But saliva has a couple of roles. One is helping break down carbohydrates when you're initially chewing up your food, but it also has mucus that helps kind of bind your food into a bolus to make a nice little slippery ball you can swallow down your gullet. And so we can actually see that other aspect of the saliva. If we take some saliva here, taking so this, part 1 of your lab talks about this. So in your lab write-up, you'll be writing up the results for those different test tubes that you did all for your pre-lab, but you also do want to include this. Use your observations from this video describing what you see, like when we add the acetic acid to the saliva and what it means. So here you're seeing me take some of Becky's spit, put it in a little spot plate here, And then I'm going to use acid to precipitate out the mucins. Those are the polysaccharides that make the mucus all slimy and yucky. Ml or so of saliva. And this is acetic acid. It's basically kind of diluted vinegar. Will actually precipitate out the mucins, the mucus part of the saliva. So now use a toothpick. We can see like, ooh, look at that, all those gooey, goopy strings there. Those are the mucins that are the other component of your saliva that are helping kind of bind and lubricate your food. So imagine trying to swallow a bunch of cracker dust. Yeah, it wouldn't really work. So this is what makes a nice slippery ball go down your esophagus. If you look in detail, totally cool gooey goop in there. All right, so that's the first part. So again, that part you should observe, write your observations, and there you can, that's what we just talked about. I showed you with genuine cracker dust, which I ended up cleaning up over break because I spewed it all over my desk. So the next part is gonna just be about the setup of the actual lab, and I'll probably go through it a little fast so you don't like sit there like boards. But I do think it's important to see how the setup works. This is for the carbohydrate lab. All right, so now we're going to describe part one of the digestion lab. Here, the salivary digestion of carbohydrates. We're going to be using... Dagnabbit! I didn't mean to do that. Oh my God, what did I do? Hold on. My apologies for spazzing on you here. All right, so now we're going to describe part one of the digestion lab. Here, the salivary digestion of carbohydrates. We're going to be using this saliva that was generously provided by my lab mate. And this will be the enzyme. And then we've got our substrate for the enzyme, our starch here. And then we can look at how this process happens under different conditions. So to start, we're going to have to just put some starch. We have 3 mL of starch that we put into each of these 3 tubes. I'm just going to put this up here. I'm going to go, there's 3 mL in there. 3 mL in there. 3 mL in there. And then also 4 and 5 are going to be controls. Part of an experiment, whenever you're using some kind of an assay, you want to validate your assay. Make sure you give your assay something where you know what the answer is supposed to be, and make sure you get the answer that you're expecting. So then you trust what the answers are when you're looking at your unknowns. So for test tubes 4 and 5, we're going to have test tube 4, which is just going to have pure starch in it. And when we do our test, it better say that, oh, it just has starch in it. Test tube 5 is where we're going to have pure maltose. I might as well just put that in right now. I will put that in a little later. And so when we can check our assay for maltose, that better say pure maltose with no starch. All right, so we have starch in our 3 unknown tubes here. Test tube 1 is going to just fill out our volume by adding 3 mL of water. So we go 1. Make sure I can see this thing. 3 mL of water. So we got 3 mL of water in test tube 1. Test tube 2, we have to add 3 mL of this quality high grade saliva. And I'm just going to bring this up to 7, so we spit it out. I'm going to spit it up into there.
For test tube 3, before we add the saliva in, we are going to increase the pH by using this concentrated hydrochloric acid. This stuff is something you got to be careful with. You should not be using this without wearing eye protection. You know, regular glasses can work. If you don't wear glasses, you got to be wearing goggles of some sort. This can damage your eyes, and your skin or your clothes or anything. So be careful with it. We're gonna add.
I should mention this is a much higher concentration of acid than we use in the protein thing. The protein lab was using like half molar. This is like almost 25 times as concentrated hydrochloric acid. So this is really like with a vengeance, like denature the amylase here.
Add 5 drops of the concentrated hydrochloric acid here. 1, 2, 3, 4, 5. And make sure, so now we have this very low pH solution, and now we're going to add 3 mils of our saliva into this. So 3 mils of saliva, but the conditions here are going to be very acidic for this salivary amylase to try to do its job. And it's important to remember, keep it in your mind now, that each of these tubes we're going to be testing both for starch using iodine, as well as testing for the presence of the products of digestion, maltose, which we're going to use Benedict's reagent for. So we're going to be putting these in the incubator. And after we're done, though, we need to make sure we take each of these tubes and we split it into 2 tubes. That way we can do the Lugol's test to look for starch in one set, and we can use the Benedict's test on the other side to test for the presence of maltose. If you forget to do this and all of a sudden put your Lugol's iodine in here without having split it ahead of time, then you're not going to be able to do the test for maltose. So at this point, we're going to take our tubes here and we're going to have to incubate them at body temperature. And we'll come back in a while and see what happens. So here we see test tubes 3, 2, and 1 are joining.
It's as part of this in vivo. It's in glass and outside of the body. We want to mimic. If it was actually inside your mouth, it would be around 37 degrees. That's body temperature in Celsius. So we're putting the condition about what it would be as if it was in your body, the right temperature.
Test tubes from parts 2 and parts 3. And we're going to close this puppy up and come back in a half an hour or so and see what happens. All right, so these have been incubating at 37 degrees C, and now we can do our assay and see what the results are. All right, so each of these needs to be split into 2 roughly equal test tubes so we can check each sample both for maltose as well as for starch. And yeah, it doesn't matter exactly, they'll just be roughly equal. And these are my 3 experimental tubes here. And then remember, I also have the control tubes I have here. This is just pure, before was pure starch. So when we do our test, this should show up positive for starch, and not showing maltose. This one here is pure maltose. So we do the test for maltose. This should be positive for maltose, but have no starch present. That's how we're going to know our test is trustworthy. So we can start with the test for starch. We use this iodine, and this stuff's going to say 3 drops of Lugol iodine. So I'm going to put 3 drops in each of these tubes. 1, 2, 3. 1, 2, 3. 1, 2, 3. Well, that's not good. Oh, that's good. 1, 2, 3. And 1, 2, 3. All right, so we look for starch. 1 is dark purple black, presence of starch. 2 is amber, that's just the color of the iodine. So this would be no starch, looks like. 3, we also have a very strong reaction here. And this is encouraging. Our pure starch is showing positive, and pure malt.
All right, so this is important. Like this could show up on your lab exam, understanding what it looks like with the Lugol's test, if there is starch or there is not starch. Here you can see when there is starch present, like in tube 1, and tube 3, which we were thinking was probably not going to break down, you can see it turns this really dramatic kind of purple black. That means yes, starch. If there is no starch, The iodine is kind of this brownish color. It just stays kind of this yellow brownish color. So it's really in here. This is the one where we had just maltose with no starch. We can see the Lugols does not react. It just stays kind of yellow brown. So make sure, in fact, when you're writing stuff in your lab notebook, you can either actually we have pictures for you. You can paste the pictures in and print them out. Actually, do that. Print them out and paste them in. But it is important that you don't just know yes or no starch, but how we got that. The lab practical tests on making sure you know the techniques in addition to kind of more of the theoretical knowledge. So this was the 5 tubes using Lugol's iodine, and then we'll do the 5 tubes using Benedict's. And again, I'm going to run this through also fast since you can just see it quickly.
Maltose is showing negative. To do the maltose test, we have to add in 4 mils of Benedict's solution into each of our other tubes. So now I'm going to, again, for each tube, let's do this up. And we'll get there's 4 in there, 4 in there, and finally 4, 2, 1. All right, so now each of these is going to have to go and get boiled for 2 minutes. So let's put these in. We have this in boiling water here. Let's also make sure, like when you're using a Bunsen burner, make sure you have a nice blue flame that's hot. If it's not adjusted, this is not hot. The water's not going to boil. You want to make sure you have the blue cone of heat. And so again, just to show you again with the starch, pure starch is going to have this very strong reaction where you get this purple-black. And in the case of the maltose, where there was no starch present, you can see the iodine just stays this kind of amber color. So this was no starch at all. This is showing presence of starch. And we'll come back in a couple of minutes when the Benedict's solutions had a chance to do this thing. All right, so these have had their 2 minutes in the boiling water. I'm going to carefully put them back in the rack here so we can see what happened. Here's our controls. 3 and 2, 1. And here we can turn off the fire. And now let's go take a look. So first we can go to our controls again, and we can see.
And here you can see that this is important to see. When it was pure starch, there's no maltose at all. It just stays blue. There's no response, no reaction. The one where it was pure maltose, we got a very strong reaction. We got this like tomato orange color here. Over here, we can see because a positive Benedict's goes anywhere from kind of green to red. So this is also where I'm moving the mouse around on tube 2. This is also a pretty dramatic positive Benedict's, meaning what is present in tube 2 here that Benedict's is telling us?
Maltose?
Absolutely. This is maltose. So this is evidence that, all right, probably the starch did break down. It's kind of what we were hoping to see or expecting to see.
This Benedict's is blue. Initially, it stays blue if there is no evidence of maltose or any of these reducing sugars. And so this is what we were hoping. This was pure starch in here without any maltose, and this shows from Benedict's that there is no maltose present because it's staying just blue. This would be a very strong positive reaction with Benedict's where it turns this reddish-orange. Again, there's a whole range of colors it can turn. It can turn from kind of greenish all the way to this kind of tomato red-orange, and that would all imply the presence of maltose in our case here. So this is great from our control perspective. Starch was negative for maltose, maltose positive for maltose. If we look at our unknowns, this was 1. This was starch just with water. So starch basically on its own after sitting at 37 degrees. Blue means, what does it mean? And why?
Right. So in fact, at this point, what I can show you, that kind of shows you how that whole thing goes. If you go to the next page, I actually have just pictures that you can look at and use to do your analysis for this part 1. This is showing the Lugol's test with tubes 1, 2, 3, 4, and 5. The Benedict's test. And yeah, if you take tube 1, it was just starch and water after an hour. Does it look like the starch broke down at all at body temperature?
No.
No, it looks like starch is pretty darn stable because the starch positive for Lugol's is still full of starch. and there is no maltose, so none of the starch broke down. Right, so this is telling us if you just look at starch, it doesn't just actually break down in front of your eyes without any help. Tube 2, does it look like there's any starch breakdown? I'm gonna do my spin the Brittany Thompson.
You wanted to know whether it's orange or what it means when it's orange?
In tube number 2, you see both Lugols and Benedicts. What do you think happened in terms of starch breakdown and why?
Well, I think it broke down because of the amylase and because the pH.
No, but I want you to, what's the physical evidence? You had your theory, but I want to say, what does the actual data tell us that we're getting from after doing the experiment? Are these things are on the screen, right? Can you see them?
Yeah.
So what do you think? What is Lugol's telling us about tube 2? What is Benedict's telling us about tube 2?
It's telling us that there is no presence of starch and that there is a presence of maltose, meaning that the starch broke down.
Exactly, right? The fact that the Lugol's is staying yellow-brown means there's no more starch anymore. The fact that the Benedict's is turning this orangey color means there is maltose there. So that is evidence. So does this support your hypothesis or not?
Yes.
Yeah, it does. This is showing like, oh my God, Becky's spit actually took the starch and turned it into simpler sugars, right? How about somebody else? What happened in tube 3, the one where we had the hydrochloric acid added to the mix?
It looks like the starch wasn't broken down at all into maltose for Benedict's.
Exactly. If you look, we still have lots of starch. Starch didn't go away and no maltose appeared. So Grace, does this support your hypothesis for tube 3 or not?
Yes, it does.
Right. So we see like a complete inactivation of the amylase in the spit. Here, Becky's spit was completely ineffective at breaking down any of the starch. Right? So, so what does this mean? Like once you swallow your food and it lands in your stomach, does the salivary amylase continue to work? Probably not. No, it's going to be acidic there. But it doesn't matter because you have lots of other amylases. We're going to have pancreatic amylase getting poured into the small intestine later. So this is just the first step of things. There'll be lots more amylase further down the line to break the starch down. And then again, we can validate our assay. We see tube 4, which was pure starch, was positive for starch, negative for maltose. Tube 5, which was pure maltose, negative for starch, positive for maltose, which tells us, okay, we can trust the results in tubes 1 to 3, where we weren't quite sure what we'd find, because when we gave it knowns, it responded in the way we expected. So again, validating your test is important to make sure you don't get trapped or don't get fooled by something. So any questions about part 1 here? Okay, let's go previous now. Let's do the lipid first, and then we'll end up on the proteins. So the lipid also has a part that is not with the test tubes. It's actually where you're using the bile to show kind of what emulsion really means. Just using oil and water and seeing how bile salts can help emulsify, can help keep the oil and water from just totally separating like they might normally. So let's play that. This is going to be Tina showing what it's like if you mix oil and water with and without bile salts and just wait a little while and see. Normally, if you mix oil and water and then wait a little while, what happens to the mixed up oil and water droplets?
They separate.
They separate, right? The classic class. I mean, that's if you ever made just oil and vinegar for put on your salad, you've probably seen this happen. All right, so let's watch what happens with when you add oil and water, but add these amphipathic bile salts, these things that bridge between polar and nonpolar.
All right, so here I'm demonstrating part 3 of our digestion lab, and the first part is to investigate the properties of bile salts and how they can emulsify water and oil.
Right, and in your lab write-up, you should describe what you see in this video. There's, a question asks, as you do this and describe things, so you obviously will describe it based on this video.
And so here I have 2 test tubes labeled A and B. I have added 3 milliliters of water and 3 milliliters of oil. And so the water is at the bottom and the oil is on top. So both test tubes have the same. What I'm going to do now, I'm going to add some bile salts into test tube B. And so you just need a little pinch. And then I'm going to use some of this paraffin, cut some squares. I'm just going to put some on top here and put some on test tube B. And then I'm going to shake.
Okay.
So we shake for 30 seconds, and then we're gonna just stop. So I'll show you test tube A and B here. And we can compare them right after I've been shaking. So test tube B, this one has bile salts in it. So we're now gonna observe what happens over the next couple of minutes and see if we can get the oil and the water to separate out.
So you can already see the bile is kind of orangey, brownish, yellowish. So you can see that's a little darker color just because of the bile salts. You can't tell from being in Zoom, but it's also kind of stinkier. Bile doesn't smell so good. You can also see there's a lot of foam and froth, right? These are basically like detergents. This is when you're like trying to cut the grease and when you're trying to soap things down. Its detergents actually act in this exact same way to get in between the oil droplets and the water and suspend the oil into the water stream so they can wash away. Right? Otherwise, you would just pour the water over a greasy plate and the grease would just stay stuck to the plate. It would never leave the plate. But, detergents are actually about getting in between the oil and the water and allowing the oil droplets to get suspended up into the water and flow off and leave the plate sparkly clean.
Which we would expect to happen in A, but let's see what happens in test tube B where we added the bile salts.
Okay, so it's almost 1 minute. So 1 minute, let's take a look at them again.
All right, so we waited a minute. You can see the water and the oil they're starting to separate here in test tube A. In test tube B, we have some foam and still an emulsification situation.
You can see the water at the very bottom. This is water that's got some of the brownish coloring from the bile salts. But all of this area in between here is actually this weird mix of tiny little oil droplets suspended in water. That's like what we call an emulsion. Here you can start, it's kind of hard to see, but you can start to see the bottom is starting to become more clear. The top is being more of that kind of vegetable oil color. And we're going to let it go a little bit longer.
So we'll wait another minute and take a look at what happens then.
All right, so we're going to take a look at our test tubes now after 2 minutes. You can see at the bottom of test tube B, we have a little water down here. It's colored a little brown, yellowish, because there are bile salts also in this water. And you can see that the test tube A, we have the water layer and the oil layer coming back. So we'll wait another minute and take a look at what happens after another minute. All right, so after 3 minutes, we can see that we have water and oil separating in test tube A, and in test tube B, we see an emulsification layer here in the middle, and then water down at the bottom, and then we have some foam up here at the top.
Right, so you can see a pretty dramatic difference. Oil and water are separating into oil on the top, water on the bottom again. In this tube, the lion's share of it is this mix of tiny little droplets that are just kind of suspended individually and still kind of just floating in within the aqueous solution. That's what we call emulsification, right? And that would allow, if there is an enzyme that's breaking apart fats, it's going to get at each of these tiny little droplets, huge surface area to be efficiently break down the fat. Whereas here, the enzyme down in the water isn't going to be able to get at most of the fat, which is floating up above it. So adding the bile salts really increases the efficiency of breaking down the fats. Even though it doesn't actually break anything down itself, it really helps the lipase do its job. In the interest of time, I don't think it's going to make sense to go through the whole. I can show you really briefly, but I think we're going to just go to the data.
There we go.
Right, she's adding in the litmus cream. This is just cream that has been mixed with this litmus powder to make it blue. Actually, I can show you, I think at the beginning. No, actually at the end, I show you. There we go.
All right, so different tubes. So
All right. Where is it?
milliliters.
So basically, it looks blue if it's neutral, if it's just cream. If it is breaking down, it will start turning red, because red is acid, and a triglyceride becomes fatty acids if you break those little bonds that are holding the fatty acids onto that glycerol backbone. So let's assume she's doing the whole experiment. You can go back and watch. It's actually it'll be fun, meditative to just watch her do it. But in the interest of making sure I don't hold you past 12:30, what I'm going to do is just go and show you pictures of what it looks like. This is what it looks like after you're done with test tubes 1, 2, 3, and 4. Test tube 4 is the one that was just the cream and water. So this was the one that had nothing happening at all. So does it look like cream spontaneously breaks down into fatty acids?
No.
No, because it's still staying just blue. There's no reddish or pinkish to it. Did it look like lipase on its own could break down the fats at all in test tube 1?
Yes.
Yeah, absolutely. It's quite pink. And this, I said, is not that surprising because cream in and of itself is actually an emulsion of fat suspended in an aqueous solution. So it actually is, helping the lipase work. But does adding bile make it even better? if we add bile and lipase, as we see in test tube 3?
Yeah, a little bit.
A little bit. It's a bit more of an intense purple pink. You can take a look. And then test tube 2 was bile salts alone with the fat. Did the bile salts alone manage to break apart the fat molecules?
No,
No. How do we know?
because it can only separate.
But how do you know? What's the evidence? What's the physical evidence that you can see that it didn't break down?
That it's still kind of blue, purple.
It's not even like purple. It's more blue, right? If it was turning purple, then it would be more evidence that there is breakdown. There's a little slightly discoloration compared to 4. I think that's more because bile salts themselves are kind of yellowish and they're going to discolor things a bit. But like if you genuinely thought this was getting more pinky purple, then you would have to say there's maybe some breakdown. If you just think it's blue, maybe with a little bit of discoloration from the kind of yellow browny bile salts, which is more what I think is going on, then there's this, it wouldn't be breakdown. But yeah, so remember, there's 2 things. We have our hypotheses with the reasons, which is important, but then we have our actual data. And then what you're going to do is look at the relationship between those and how much does the data support your hypotheses or not. And the data is more of these assays looking at the breakdown or not. So that's the carbohydrates, that's the lipids. For the proteins,
Using for
that is what the little square of egg white looks like. We basically cut the egg white into 5 squares, as much as possible, exactly the same size. So then after an hour, we can look to see, is the egg white the same size or is it smaller? Would this be a qualitative or quantitative kind of assay?
Qualitative.
Qualitative. We can tell if there's more or less breakdown, but we can't tell exactly how much. And basically, we are going to put the egg white squares in the 5 things, and then we're going to add in some with acid, some with base, some with pepsin, some without pepsin. And we did this, and I'm not quite sure what went wrong. It could have been something wrong with the reagents, but I can say that you're not going to see a picture. Like if you go into the next part, the results. There's pictures for the carbohydrate. There's pictures for the lipids. We didn't give you pictures for the proteins because they all look the same. It didn't look like anything. So, but that's something you can write up in your lab book, is like, what you expect, what you found, and you can talk about like what might have gone wrong. Lots of things might have gone wrong. Actually, so the last thing I will show you here. Oh, come on. There it is. This is a spreadsheet that's just available at the bottom of that same page with all those pictures. And this is just showing across a bunch of different groups, where 3 is like 3 pluses. This is just showing across many groups where you want to kind of make sure that everybody's getting the same results, because doing it once is suggestive, but if you do it again and again and keep getting the same result, then you trust it a lot more. Here we can see tube 1 again and again. People found starch was there with no maltose. The one with spit and starch, there was, no starch left, but maltose showing up. The one with the acid, there was still starch and no maltose. So this is actually useful. Being able to have this data to show in your write-up that your observations from the one that we did in the lab is supported by this greater group data as well, right? Because you don't want to just do something once and say, I got the answer. You want to make sure if you do it again and again, you keep getting the same answer before you really trust it. For the protein one, you can say, from this data, it looks like in general, maybe there is would this make sense that tube 3 had the best digestion?
Yes.
Right, that was the one that mimicked the stomach the best, had the acidic environment where pepsin is going to be optimized. But I have to say, this is not convincing. The carbs one is very convincing. It's exactly what we expect, and everybody got it. The protein one is like, yeah. And then finally, lipids, right? This also is pretty convincing. Tube 3 had the greatest breakdown where we had the bile and the lipase. Tube 2, which was just lipase, still had breakdown, but not quite as dramatic. And bile alone, or just cream on its own, or I should say bile and cream without the enzyme, or just cream just sitting alone, doesn't really do much of anything. Right, so this spreadsheet will be useful in your write-up. You want to use this data to support your, the observations that we actually got from that one time we did it in the lab. You want to say, yes, we found that there, and that agrees with the greater data that we see across the many different groups. And again, that spreadsheet is right here at the bottom. It's linking on this page, Digestion Lab results. There's pictures of the carbs, pictures of the lipids, and that spreadsheet is just down here at the bottom. Right, and then the next page is just, submit to Digestion Lab.